All right, so my name is Bram, and I'm a postdoctoral fellow working at the University of Cambridge in the unit of cardiovascular epidemiology. And at the end of this afternoon, I just wanted to give a really lighthearted presentation about the background of this tool and why we developed this. I'm going to go quickly through a couple of slides because some people need to leave at 3 o'clock, but it's easy because some of the things were actually repeated in the previous talk by Dan, by SomaLogic. So, as you were told before, there's a major challenge in drug developments, and the two major things were high failure rates, and particularly in phase 2, and the identification of new targets. So the first leads to an attrition problem, and the second is actually an innovation problem. So this figure comes from a really nice paper, which all of you recommend to read, which tells you about what actually had made projects successful in AstraZeneca. So if you look at the phases here, from preclinical to phase 3, you see the least successful discoveries were actually in phase 2. A lot of projects failed here. So you can ask the question, why is that? Why projects were closed in phase 2? So we can look at the right graph over here. So in preclinical and phase 1, basically drugs failed because of a safety issue. But in later stages, in particular phase 2, there was actually an efficacy problem. So what's the problem of this lack of clinical efficacy? There's a couple of reasons, but the major one is actually that there's missing targets linkage to the disease. And if you look at the efficacy again, so in phase 2 projects, which ones were the most successful? So those that had a genetic linkage to the target of the disease. I'm going really quickly now. So this is, in essence, what was told to you today and what we're trying to do. So we're trying to develop genetic targets for drugs, and how genetics can help to find the right target is actually by trying to find a link between the targets and disease. So we're trying to find a proper target by finding the genetic difference between a healthy population and disease population. So there's 3 very simple steps. So first you try to map the genetic differences between the healthy part of the population and disease population. Then you try to understand how these differences lead to the disease. And then the third step is to actually find a drug target that can actually reverse this entire process in the population. So, a lot of you have heard about GWAS, probably at the conference as well. It's GWAS, everything is GWAS. But it's a really useful tool still to actually identify drug targets. So, for those of you that are not familiar with GWAS, basically it's a method that allows you to find associations, genetic associations to disease, where we interrogate all the millions of variants across the entire genome. Now, there were a lot of important developments that were lay ground to this particular technique. So, as a completion of the Human Genome Project, we had fine mapping of genetic differences by projects, for example, like HapMap, the lowering costs, of course, of these experiments, and the development of high-throughput methods. So that's basically GWAS in a nutshell. So, Dan showed you before that we can actually mimic a drug response curve through the build-up of a response curve by using genetic variants. And these genetic variants you obtain basically from GWAS analysis, so you can use summary statistics. That's the whole key idea for all these data being available. So you can build this DROS response curve. So in this case, we do a lookup for molecular trait levels. So, for example, the CRP protein that we are looking up. There's a different range of effects for different variants. So you have loss of function variants with very small effects, but also you have gain of function variants with a big effect on the disease and on the protein. So this is basically what we're trying to build. Now, proteins are ideal intermediates for building such a dose response curve. So first of all, because they're basically the closest to the DNA, so they're on a much more proximal control. Secondly, they're the biological effector. And thirdly, also, they're the typical target for in drug development. Right. So how do you build a genetic dose response curve? For example, to do your Mendelian randomization analysis. So you use, as I said, the summary statistics from GWAS. And you simply need just the summary statistics for your exposure, so your protein, being CRP, IL-6, and so on, and summary statistics for disease you're interested in. And then you can build this particular graph, so the response curve, as you see on the right. But there's two things we actually need to pay attention to. Is first of all that the variants that you will use do not violate assumptions of Mendelian randomization. So as Dan, for example, said, if you look at CRP, you only should look at variants that only influence CRP through the CRP gene itself, not using any other intermediates. And secondly, also, you should have what's called sufficient statistical power to actually detect an effect, which in practice translates to that these variants should come from measurements in really large sample sizes. So this is actually the start of my PhD. So I had to look up summary statistics for a lot of analysis, and this was also the most frustrating part of my PhD. So I was trying to build these curves, and how do I get this data? I actually had to go to papers, copying and pasting from tables and trying to basically collate all the data to do my analysis. Even worse cases, sometimes data wasn't published, so I have to write an email. Say, oh, dear Mr. Scientist, can I please have a lookup for these and these variants? Can you give me the summary statistics? In exchange, we will give you authorship and a consortium benefit, because as you know, in the academic world, our currency is basically authorship. So that was the beginning of my PhD, and I had to do it for a lot of different diseases. So then there was some protests actually in literature, a lot of editorials in Nature and in PLOS Genetics that basically were telling, so wait a minute, so you're doing all these massive studies and you have lots of data available which are really useful for repurposing, for example, for the Mendelian randomization analysis that we're interested in, but you're not publishing this data and your studies are funded by public funding. So from now on in the future, we really like to ask you almost forcefully to actually make your data available as soon as you make the publication available. So there's, for example, a rule, as I understand from NIH, as soon as there's a co-author on a paper from a certain GWAS study, that basically that study should make the GWAS data available. So there was some incentive actually to make data more publicly available. Now, luckily for us, there's lots of data that we can look at. So this is a graph that tells you how many GWAS studies were done. So back in 2008, it was less than 100, but when we're entering 2016, we actually have thousands of studies done for a lot of phenotypes. With that, we actually can look up summary statistics for the analysis that we want to do. Also, we're lucky in the sense that sample sizes over the years also increased. So in the beginning, the biggest sample size you could find was roughly 10,000 people in total. But now you easily have sample size over 200,000, I believe. The largest study you actually can find is for GIANT, which is a consortium that works on anthropomorphic traits. It's almost 1 million size. So that means we have really good instruments, very powerful instruments statistically to do the analysis that we care about. So this is approximately how the situation looks now. Looks like just before PhenoScanner was published. So you want some data, how do you get it? So you can go to individual studies. The problem is, however, they typically look at just a small number of phenotypes. But you can get all data, so the original genotype and phenotype data, but the numbers are small. So the instruments that you will use from these studies are usually not so powerful. Some consortia, so based on, for example, the papers that I just showed before, actually make their data publicly available. So you can get genome-wide statistics for, let's say, more than 3 million variants, and you can just download them directly from the web. But the problem with consortia, again, is typically they're just focused on one phenotype. So if you want to do a PhenoScan, so do a lookup across many different types of disease and traits, you have to go to many different websites to get all your data. Another option is the GWAS catalog. So basically the GWAS catalog is a collation of all the tables that you find in papers about GWAS collated into one data set. There's actually a person hired for this in the European Bioinformatics Institute that does nothing else than actually copying and pasting tables and formatting in the right sense in order to download your data. So it's good, so you can get data for many phenotypes, but the problem is you only can get top hits. So it's not genome-wide, but the numbers are big. So these different scenarios, still, it was difficult to actually obtain your data. So the problem basically with all these different databases, that's of course, first you have a lot of summary statistics available, but they're decentralized and they're unharmonized. So unharmonized basically means they use different genetic coordinate systems. They use different variant definitions. In the GWAS catalog, there's the occasional mistake, so you can't trust the data and you have too many places to go to and do so many conversions. So you want to make it easy. So you want to do, you actually want to have a tool that can do everything for you with a very simple mouse click. And this is why a former PhD student in our group has developed PhenoScanner. so which is an online tool that contains the summary statistics from all publicly available data. You simply surf to the site and everything is under your hands. It now contains data for more than 1,000 diseases and traits for more than 70 million SNPs, and it even can calculate proxy variants for you. So proxy variants are variants that are very similar to the variants that you look up, and if something is not available in a certain data set for the exact SNP that you're looking up, basically it can calculate and find the data for a certain proxy for you. So it's a really helpful tool. Now, this is how it looks like. You surf to the website, you enter your SNP, you tell what kind of catalog or data set you want to do the lookup for, and there's a number of settings, so how strong I want the p-values to be. You can select what kind of proxies you want. You press the button and the data comes out. Or you can do it in batch, so basically uploading a small file with your set of variants, and then you can get the data as you like it. So how does it work? And as I said, you just take a SNP, ask the website to calculate the proxy, you get all your associations, and then you can use the statistics for whatever you want. So for example, for Mendelian randomization, where you try to do the calculations for your intermediates and then for your outcomes. So here, for example, for HDL, triglycerides, height, and your outcomes, coronary heart disease and rheumatoid arthritis. So this is how typically you can do a very simple lookup. Now, actually, the original idea for this session was to do an actual workshop. So pretending we're in school and going to press some buttons and entering some numbers. But for the sake of time, and I think it's also very difficult to read variants and text for the people in the back, I'm just going to directly give you answers. So my plan was actually to do a very simple demonstration. So finding SNPs that are useful for drug target evaluation, and then finding indications for repurposing drugs, and then evaluating SNPs, whether we can use them in Mendelian randomization. So let's pretend I've done an analysis. So let's say I use the SomaLogic platform and I know which variants actually influenced my levels of IL-6. And IL-6 is interleukin-6. It's a protein that's involved in inflammatory response. And let's say we already know that, for example, it's involved in a disease, coronary artery disease or type 2 diabetes. So I have some summary statistics. So this is how Manhattan plots look like. So this basically tells me I found two signals with two top variants that influence my levels of IL-6. And this is how my summary statistics looks like. So I have a variant on chromosome 1, one on chromosome 6. These are the names or indices of the variants, and I have summary statistics. So the typical summary statistics you use for MR are the effect size and the standard error. So we're going to use these variants, and now we're going to pretend we're going to have the interactive workshop. So I would ask you to go to a website called snppedia.com and check whether these SNPs are in genes, because we need some instruments for our Mendelian randomization, but also we need a drug target in order to develop a potential drug for. So the key idea for this exercise basically was, okay, I have two SNPs, one of them is not in a gene, and typically for the practical I would assume that a gene encodes for the protein, which is the actual target that we would develop our drug for. So which of these SNPs lies in a gene? And the answer would be, so the first SNP, and it lies in IL-6R. So this is actually the example that Dan also gave, so the IL-6R receptor. So then the next question is, we just wonder, so is there already a drug developed against this target? So there's this really nice database. I recommend all of you to actually use this database. It's called the Drug Gene Interaction Database. So if you would surf to this website, basically, you can enter the name of this particular gene and you would find a drug target. So you actually would find two. Maybe I can just, because of course I've done all the exercises myself already. So let me see. So this is how the website looks like. It's very easy. You just enter your gene, then then the targets come out, and you see the drug that was shown by Dan before as well, so tocilizumab. And if you click on this, you see there's a couple of indications. So yes, there is a drug already developed for these targets, and it's indicated for rheumatoid arthritis and juvenile idiopathic arthritis. So that's your answer right there. So there is already a drug target, and it already is indicated for certain diseases. Right. Now we want to know if our SNP is actually associated with other diseases. So for this, we actually use a PhenoScanner. So I would ask you to go to PhenoScanner, enter our SNP name, and then do the particular query with these settings. The idea is that we already know that this particular SNP then is involved in rheumatoid arthritis. There is a drug already developed for this disease. So the idea is that maybe you can actually repurpose this drug, but then for another disease, which is why we're going to do the lookup. So we're going to use our original SNP. Then we would press search. And then a couple of questions. So is our SNP or our proxy associated with the disease that we found for? So the results actually come to you in an, let me see if it comes, in an Excel file. So I have opened this. So don't be flabbergasted by the output. So you get a lot of different data. So lots of columns. So horizontal lines are variants, and then you see the position and the alleles and a lot of things that are not so important for this talk right now. But one of the columns is trait, and it tells you for which this particular SNP has been found. So it's involved in levels of C-reactive protein. We find the disease back for which we found this drug, so rheumatoid arthritis. But if you go down, it's also, this particular SNP is also involved in asthma, and if you actually go further down, also in coronary heart disease. So the hunch you would get is, well, maybe this would be a useful drug actually that we could repurpose for coronary heart disease, just to get an idea. So the question is, could we then actually design a genetic instrument for Mendelian randomization to use IL-6 levels, because we know our SNP influences IL-6 levels and we want to actually develop a drug target. So if you want to do a Mendelian randomization analysis, I told you, you need these summary statistics, effect size and standard error. And if you actually go back to the Excel file, you will see there's no betas and no SEs in this particular lookup. So in this particular instance, these studies did not supply the effect sizes, which I'll come back to later. There was a bonus question as well. So we have summary statistics for our SNP in IL-6R. Are they good instruments for Mendelian randomization? So Dan already has explained this a little bit. So the key idea is that one of the key assumptions for Mendelian randomization is that your SNPs should only have an effect on your disease of interest only through this particular protein and nothing else. And if you actually look at the Excel table, you see that this SNP not only influences IL-6 levels, but also soluble IL-6R levels. It influences CRP levels. It influences fibrinogen levels. So it will be a really bad instrument to do a formal Mendelian randomization for. So these are a couple of purposes for which you can use PhenoScanner. So what we actually just had done in this practical exercise is looking at this signaling model. I think Dan also demonstrated this. So we looked at a particular SNP that's involved in the IL-6 receptor. So what normally happens is IL-6 binds to the receptor, and then there's a classical signaling cascade that's further downstream. And this SNP, particular SNP, disrupts this signaling process. So if you collate the data that comes out of PhenoScanner, this is how it looks like. So conventional risk factors for this particular SNP, not associated, they're on the no. But inflammatory factors, so for example, soluble IL-6R, also the interleukin-6, which we took as an example, are changing for each extra copy of the allele for the minor allele of this variant. So the more copies you get for this particular variant, the higher are the soluble IL-6R levels or interleukin-6 levels, but the lower C-reactive protein and the lower levels for fibrinogen. And the same lookup also tells you that for the same change for your variant, so going to the minor allele, that risk decreases for coronary heart disease, rheumatoid arthritis, but interestingly, also increases for asthma. So you could get a hunch that, okay, maybe if you give a certain person a drug that mimics this effect, it will lower your chances for coronary heart disease, but there could be some side effects. Perhaps you develop asthma. So it's also a way to actually evaluate off-targets for drugs that you're interested in. So just to show you, so this is the summary data that we have looked up in PhenoScanner, so for this particular variant. And actually, if you look at So for each copy of this minor allele, you see that the levels increase for soluble IL-6R and interleukin-6 and decrease for C-reactive protein, fibrinogen. The drug actually that we're looking at has the exact same directions of effects. So this is a change for tocilizumab, pre and post change. And you see for soluble IL-6R, it goes up, and then interleukin-6 goes up, like each extra copy that you get for this particular minor allele of this variant, and CRP goes down and fibrinogen. So this is a classical example that you could actually look up using PhenoScanner, but in a much more easy way now. And this has been published in many different studies. So this particular example is particularly in the Lancet, which also published a paper called, so it had Mendelian randomization for IL-6R or something like that in the title. And actually, if you read the text, you should not consider this as a Mendelian randomization, exactly because they violate the principle and assumptions of Mendelian randomization. So that was my original plan for the demo. It's important to know that we're still working on PhenoScanner. Soon there will be a new version released. So what we have done actually today and demonstrate today is that you can do an individual query. You go to the website and it has a large underlying database that does the lookup. But one of the things that we have to do externally is to actually look up the genomes, for example, right? So it will be handy to have this. So we're going to add a lot of annotations. And also we're going to collate the phenotypes. Sometimes if you collect data, phenotypes don't exactly have the same name. So for example, you have coronary heart disease, coronary artery disease, and so on. So we are going to actually apply some ontologies so you actually have much more unified and clear data. So we're looking at the same things. Another thing that we also noticed is that so for IL-6 levels, we actually could not find any summary statistics. And then if you look at this catalog, you actually will find, okay, there's a lot of diseases looked at, but there's a fairly limited amount of proteins for which there's genetic data available. So nowadays proteomic studies are coming up, like for the SomaLogic, but there's not too many out there, especially not present in public databases. So we have a lack of genetic instruments, like for example, variants that influence protein levels, for which they can help us actually to do these Mendelian randomization analysis. So with this and many other things in mind, I think our group has set up this very large data resource, so the interval cohort. We have lots of data available that basically allow you to go entirely from the DNA to your final phenotype. We have whole genome sequence data, so a very fine resolution of the genetic map. We actually have genotype data for all individuals and anything in between. So we have measured protein levels, lipids, metabolites, all which could be really good instruments for these causal inferences from Mendelian randomization analysis. Now, just a little bit of detail. So what are these intermediates that we have? So we have, for example, lipid species, metabolites, more lipoproteins from brain shake. So I work a lot with Olink data, which is a limited proteomics platform. Each platform is specific for a certain, what would you call, like a group of diseases. So there's the cardiovascular platform, there's the neurology platform, and so on. And also we have SomaLogic data, actually, for which we have done a GWAS. So we have a PhD student in our group, Ben Sun, that actually did all the analysis. So SomaLogic, I think you already kindly introduced it. So it's a highly specific platform for a large set of diverse proteins with certain focuses. So basically they have a bias towards clinical and pharmaceutical targets. So the analysis were published, or actually I can't say published, so put on a preprint server called bioRxiv. And currently is under review in Nature. So a brief summary, so just to show you how powerful this platform is and what also will be added to PhenoScanner in a later stage. So we've done analysis of more than 3,000 proteins in more than 3,000 individuals, and we found 2,000 association signals, which is staggering. So we find, compared to other proteomics platforms, well, at the time when we did the calculation, 10-fold more associations as compared to everything that had been done before, which is really an amazing feat. And also, the other good news is that for many proteins, we now explain 10% of variance. So it means, in statistical terms, that you can, when you explain a lot of variance, that these can be potentially really powerful instruments to do your Mendelian randomization analysis, for example. Now, Full disclosure, I have to be honest, of course. There's other developments going on that are similar to ours. A really important one, and I definitely would like to ask you to check this out, it's called MR-based. So they have a similar model like PhenoScanner, but also what you can do in addition is actually to do the Mendelian randomization analysis online. So you enter your variants, you get a lookup, and then you can perform the Mendelian randomization analysis just online. So it's not high throughput, but if you want to have a quick gist of what you can do with your variants, go to this website and definitely try it out. Also, actually, I was trying to write a ground with an idea because I was learning through Coursera machine learning. I thought it was really useful in the future. I thought, maybe you can use machine learning to Mendelian randomization. And behold, just 2 weeks ago, somebody published this method, completely worked out with the god of Mendelian randomization, so which is George David Smith, and put this on the preprint server. Automated Mendelian randomization. So basically everything is out of our hands now, and I felt like, okay, I can pack out my suitcase and go home. So good paper to read. It also tells you how careful you should be in instrumental design. So they use machine learning to design optimal instruments, but you still need the human eye. For example, there's a causal association, according to this paper, between let's say it was in intelligence, education levels and birth weight, but in that order. So the higher your education levels typically is, the higher your birth weight will be. And of course, everybody understands that it doesn't work that way. But it's a really interesting development, and I think you definitely should read this paper. So, quick summary. Well, I mean, we've discussed everything already, but PhenoScanner, so I believe I'm using it sometimes as well for my analysis. I think it's an easy and powerful tool to use to do phenome scans. We're still trying to address some issues or some extra benefits and features that the database should have. So like, for example, the annotation. We're also trying to work on automated interrogation of the database. So there's one person in my department, Stephen Burgess, who's a brilliant scientist, who's actually developing an R package. So R is a statistical language that allows you in the future to do automated analysis by yourself. So no more pressing buttons, you just can program whatever you want. And of course, by looking at interval data, so we have a really great resource available, which is the interval study, which we have a lot of intermediates, so providing basically a really good genetic toolkit to do the analysis that we're interested in. So there were a couple of questions I understand whether the SomaLogic data will be available. So I talked to our PI, and as I understand, so the data, as soon as the paper is actually accepted, the SomaLogic data will be all available, all associations genome-wide in the PhenoScanner database for future use. I think that's important to mention. Yeah, that's it. I made it in 30 minutes, so I hope we're a bit on time. These are the people I'd like to acknowledge. So this is basically the group I work in, and some external people like our expert Robert Lange and the people from Merck. That's it. Thank you. If you have questions? Sure.
For my education, how do you factor copy number variation into this? Can you use copy number variation and SNPs within those, I don't know, amplified regions as a dosing proxy? Is that the right terminology?
Yes, it is. And the honest answer is, oh, no, I can yell as well. That's good at it. So the honest answer is basically we don't look at copy number variation. Typically we use single SNPs to do these analysis. But I mean, it would be something worthwhile, right, to look at structural variants like in this area called 11q13. You have copy number variations where you see giant peaks basically of multiple. But the problem with these structural variants is that they're very difficult to analyze. Because it's not always exact copies that you get. Sometimes it's half a gene, sometimes they're overlapping. So it's very difficult, I would imagine, to actually build a dose-response curve on this. And the second problem is that structural variants sometimes are very difficult to call as well. So it's a color, basically a genotype color dependent problem, I presume.
Another question.
Yes, please.
Okay. So with SomaScan and the type of interaction that I showed earlier, we're measuring the availability of an epitope, okay, not the concentration of a protein. Everyone thinks fold change. I think with O-link, you'd probably be measuring protein concentration changes. But for us, I'm pretty sure you're measuring epitope availability,
Sure.
which could actually be influenced by things like receptor ligand affinity, and your SomaMer is competing for that interaction.
Sure.
How do you factor that into? Is that factored in any way to Mendelian randomization as an approach?
So for the method itself, So for the method itself, as far as aware, no. What we do look for is that the variants that we find are truly cis-specific. So for example, with O-link data, sometimes we're not so sure about the probe if we only see a trans association. So we try to correlate the data with SomaLogic to get an idea. That's about the only thing you basically can do. But specificity for us is really a major thing. So it's definitely something we always look at before we do any follow-up analysis with Mendelian randomization. Does it answer your question?
So you have to take care.
Yes, very much.